Hello viewers, welcome to my channel. Today I will talk about a very important topic for all chemistry paper exams, and the topic is synthesis and applications of 10 heterocycle based drug molecules. Here on the screen you can see the names of 10 heterocycles that I will discuss today, their synthesis and applications obviously, and the structures as well, where I have highlighted the basic cores of heterocycle of the molecule. So first I will talk about the extensive list of the drug molecules, very minimal information regarding their usage, and then finally I will come one by one regarding the synthesis and applications of all the drug molecules. So ranitidine comes first, and it is a furan based drug molecule and commonly used in the treatment of peptic ulcer disease, gastroesophageal reflux disease. Suprofan, this is a thiophene based drug molecule and it is a non-steroidal anti-inflammatory drug used for reducing pain and inflammation in the eyes, including glaucoma. Atorvastatin, very important and known to the people who are having the high blood pressure because it is an inhibitor of HMG-CoA reductase and also acts as anti-hyperlipidemic. Molindone, this is an antipsychotic drug and used to treat the chronic schizophrenia. Moxifloxacin, this is used to treat certain infections caused by bacteria such as pneumonia and also for skin diseases and abdominal infections. Hydroxychloroquine, this is quite known for us for last couple of months because of the COVID-19 treatments and used to prevent or treat malaria caused by mosquito bites. Papaverin, this is a very important drug molecule and it improves the blood flow in patients with circulation problems. Phenobarbital, used to control seizures and also to relieve anxiety among the patients. Metronidazole, quite known to us, and it treats and prevents bacterial and parasitic infections. And finally, the last of the list is diazepam, and this is used to treat anxiety, muscle spasms, and fits. So now we will move into the synthesis and applications of individual drug molecules. First comes ranitidine. It is a medication that decreases stomach acid production and was discovered in England in 1976 way back and came into commercial use in 1981. And this is on the list of WHO's essential medicine. Commonly used in treatment of peptic ulcer disease and gastroesophageal reflux disease and also in Zollinger-Ellison syndrome. However, it can be taken by mouth, injection into a muscle or injection into a vein, whatever this depending on the situation of the patient. So the synthesis started with furfural or furan 2 aldehyde, and when the furfural is reduced to by means of NaBH4 to furfuryl alcohol, using also lithium aluminum hydride can also be used as the reducing agent. It will give the furfuryl alcohol. Now the furfuryl alcohol reacts with dimethylamine in presence of formaldehyde at room temperature to produce the 5-dimethyl-aminomethyl derivative of the furfuryl alcohol, which on further reaction with 2-amino-ethanediol will produce eventually the thioethanamine derivative. Okay, and then this thioethanamine derivative, when reacts with nitroethanamine molecule in a nucleophilic substitution reaction, eventually it will produce ranitidine molecule, which is of our concerned drug. Next in the list is Suprofan. Suprofan is used exclusively as a topical ophthalmic solution, typically to prevent miosis during and after ophthalmic surgery. It is a non-steroidal anti-inflammatory drug and usually prepared by one-pot Suzuki carbonylation hydroxycarbonylation sequence. The synthetic approach involves 2-bromothiophene and 4-vinyl phenylboronic acid as the initial substrates and when they react in presence of palladium catalyst and external source of carbon monoxide and a base tetramethyl ethylene diamine, commonly known as TMEDA, it produces thiophenyl ketone. That one. So this phenyl thiophenyl ketone under similar set of reaction conditions only in presence of change in media, that is going from basic, now it goes to the acidic media, that is in presence of HCl, under the similar kind of conditions, to give the 2-aryl carbonyl propionic acid derivative, which is commercially known as Suprofan. So this is the synthetic strategy for Suprofan. Atorvastatin was first commercialized as Lipitor by the company Pfizer in 1996. Lipitor broke records by becoming the world's best-seller pharmaceutical of all time. So you can guess how important is this drug molecule. Now, atorvastatin is an inhibitor of HMG-CoA reductase and acts as anti-hyperlipidemic and most prescribed drug due to its lower incidence of serious side effects. Now, the synthetic strategy of atorvastatin involves a multi-step process. It's a quite large molecule in that sense compared to Suprofan or Ranitidine, that one. And it initiates with the reaction of oxopentanoate ester derivative and aniline to produce the amide derivative, that one. Now, this amide derivative upon reaction with benzaldehyde under acidic condition first gives benzylidine phenyl pentamide derivative. You see here. Now, this pentamide derivative on further reaction in turn with fluoroamide derivative gives the fluoroamide derivative on reaction with parafluorobenzaldehyde. Now this 1,4-dicarbonyl derivative that we obtained right now on further reaction with the diol protected amino ester, which you can see above the arrow in presence of pivalic acid, generated the pyrrole ring structure upon cyclization. And finally, the acidic deprotection of this pyrrole structure gives atorvastatin as the final target drug molecule. Next one is molindone. Molindone is a more active antipsychotic drug molecule than chlorpromazine, a quite renowned drug molecule, and its sedative effect is less expressed compared to chlorpromazine. It facilitates the reduction of spontaneous movement and aggressiveness in the patients, and that's why it is used for the treatment of psychotic disturbance, particularly in case of chronic and severe schizophrenia. Now, the synthetic strategy of melindone first initiated with the nitrization of dialkyl ketone using nitric acid or methyl nitrite. Now, here the dialkyl ketone is specifically to be mentioned as pentane 3-one. Then it will give the nitrosodiethyl ketone as the product, which you can see over here. Now this product on reduction with zinc and acetic acid will lead eventually to the formation of 2-amino diethyl ketone, and then further reaction with cyclohexane 1,3-dione will give the indole derivative. This indole derivative on further reaction and aminomethylation using morpholine and formaldehyde will give melindone as the target drug molecule. So this is the overall synthetic route for melindone. Next come moxifloxacin, a very important drug, and it is an antibiotic used to treat number of bacterial infections like pneumonia, conjunctivitis, endocarditis, tuberculosis, sinusitis. So you can imagine the wide range of application of moxifloxacin. Now it belongs to the fluoroquinolone family of medication. That means you understand that it will have a quinoline structure into its molecular skeleton, and the usual function results in bacterial death through blocking their ability to duplicate the DNA. The synthesis begins with the trifluoromethoxybenzoyl chloride and potassium oxopropanoate ester under basic condition to afford the trifluoropropanoate derivative. This one. Now this trifluoropropanoate derivative on further reaction with triethoxymethane and acetic anhydride, it will give the trifluorobenzoyl acrylate, and then this benzoyl acrylate on reaction with cyclopropamine will end up with cyclopropaminobenzyl acrylate. Okay. Now these need to be cyclized, and this cyclization to pyridine fused ring structure takes place in presence of a base, usually sodium hydride, and DMF is preferred as the solvent, and it needs an acidic workup. And then the overall intramolecular base catalyzed nucleophilic aromatic substitution is used to prepare the bicyclic ring system of highly substituted aromatic compound. A nucleophilic aromatic substitution reaction is necessary and used to introduce the saturated piperidinopyrrolidine, which is used as the appendage to furnish moxifloxacin as the final product. Then comes hydroxychloroquine. Hydroxychloroquine is an antimalarial drug used to prevent or treat malaria. This medication is also used to treat certain autoimmune diseases, which include the rheumatoid arthritis, you can also see. And it belongs to a class of medications known as disease-modifying anti-rheumatic drugs. In short, DMARD. It can reduce the skin problems in lupus and prevent the swelling or pain which occur in case of arthritis. Now, for the synthetic strategy or the synthetic approach, the chloropentanone on reaction with the amino alcohol in presence of transition metal catalysts like cobalt nitrate and nickel nitrate, supported by magnesium and calcium oxide, it gives the hydroxyethyl aminopentanone derivative. That one. Further reduction of this carbonyl group using ammonia and ranitidine, and which will eventually, in presence of hydrogen, of course, generates the aliphatic amino alcohol derivative. This amino alcohol derivative then participate in a aromatic nucleophilic substitution reaction with 4,7-dichloroquinoline to give the hydroxychloroquine. That is the final molecule. Now, one thing need to be mentioned here, that it has been studied for an ability of hydroxychloroquine to prevent and treat the COVID-19, but we need to know that the clinical trials found it as ineffective and possible risks of dangerous side effects. So this is not anymore hydroxychloroquine is prescribed for the COVID-19 treatment. Next one is Papaverin. This is an opium alkaloid antispasmodic drug, and it is a vasodilator and used to improve the blood flow in patients with circulation problems, and works by relaxing the blood vessels so that the blood can flow more easily to the heart and from heart through the body for the human beings. And this is used to treat many conditions that cause spasm of smooth muscle, and this includes the chest pain, the circulation problems, heart attack, or disorders of the stomach or gallbladder. Now, we will talk about the synthetic strategy now. The dimethoxyacetophenone, you can see the substrate of choice on the first, on reaction with the nitroalkane gives the oxime derivative, this one, and which on subsequent reduction by means of zinc chloride will produce the amino carbonyl compound. Okay, so the dimethoxyacetyl chloride further reacted with the amino carbonyl to produce the acetamide derivative, and this on reduction by sodium amalgam in aqueous media reduce the keto group of the keto group to a secondary alcohol, which is very known to us, very common chemical strategy for that. And finally, the reaction of phosphorus pentoxide facilitates the cyclization followed by aromatization to furnish papaverin as the final product. Okay, and it contains, as you can see, the isoquinoline ring structure. So this is the overall synthetic strategy for the drug molecule papaverin. Next, we will talk about phenobarbital. Phenobarbital is a medication of the barbiturate type known by the trade name Luminal. It is used both as a sedative and also as hypnotic. Also useful in control of epilepsy and can be given to children for treating convulsions. It may be used intravenously, injected into a muscle, or may be prescribed to be taken by mouth depending on the condition of the patient, and also as prescribed by the doctor. The synthetic approach utilizes the diethyl carbonate in presence of a strong base, which is sodium ethoxide, can be, to give the alpha phenyl cyanoester derivative. Okay, so this alpha phenyl cyanoester derivative on alkylation using the ethyl bromide will eventually give the alkylated product and proceeds via a nitrile amination intermediate to give the alpha phenyl alpha ethyl cyanoester compound or derivative. This on further reaction with converted in, or this will finally converted into the 4-amino derivative upon condensation with urea. So this is the 4-amino derivative. And finally, this 4-amino derivative need to be hydrolyzed. So in order to get the phenobarbital, and you can see this imine got hydrolyzed to the ketone here, and giving the final molecule phenobarbital as the synthetic product. Next one is metronidazole, quite known to us and very important drug molecule. It is an antibiotic and antiprotozoal medication, and it falls in a class of medications called nitroimidazole antimicrobials and works by stopping the growth of bacteria. It is available or can be taken by mouth, used as a cream, or sometimes need to be injected into a vein depending upon the seriousness of the patient and as prescribed by the practitioner, medical practitioner. Now, the key intermediate to the synthesis of metronidazole is 2-methyl imidazole, as you can see on the screen, and which can be obtained in two ways, either from the reaction of glyoxal, ammonia and acetaldehyde followed by cyclization, or the other way around with or from ethylene diamine with subsequent steps like acetylation, which will be followed by cyclization in both ways. Now, having this 2-methyl imidazole synthesized in either way, this on nitration by means of nitric acid and sulfuric acid gives 2-methyl 5-nitroimidazole as the product. And then this one on N-alkylation with 2-chloroethanol in alkaline medium will lead to metronidazole as the target molecule. Now, the last drug molecule of the extensive list is diazepam. Diazepam, first marketed as Valium, and it is a medicine of the benzodiazepine family that acts as an anxiolytic. It controls convulsions in status epilepticus. However, it is not used to treat epilepsy as there is a rapid development of tolerance as found in patients. It is used to treat anxiety disorder, insomnia, withdrawal effects of alcohols. It can be taken by mouth, injected into muscle or vein, or can be used as a nasal spray. So these are more or less the applications of diazepam. Now we will move into the synthesis of diazepam molecule. The first step in the synthetic approach of diazepam is a double acylation of an aromatic amine. Yes. First, by nucleophilic acyl substitution of an acid chloride by the NH2 group, and then by the electrophilic aromatic substitution of the ring. The resulting intermediate is then trapped by the second equivalent of the parachloroaniline, and that will lead to a six-membered ring structure with two nitrogen atoms, which is the dimer, so to say. Now the hydrolysis of this dimer reverse the cyclization and frees the simple electrophilic substitution product, as you can see over here, in 2 moles of quantity. And then a 7-membered ring is then produced, as you see over here, by the intermolecular interaction with an amino ester compound. This cyclization takes place by amide formation between the ring NH2 group and the ester group of the co-reactant, and by imidation of the ketone group by the primary amine. Now, with the final methylation of the amide NH group by dimethyl sulfate in presence of dimethyl carbonate, it will complete the synthesis and will produce diazepam as the target molecule. So thank you viewers for watching my channel TG Chemistry, and keep